was really interesting, Bill, was that the recovery of a single specimen from the Ilium vent site was really important to establishing part of those relationships.
Wow.
He just ran away.
So it was very hard to get the DNA out of that single specimen, but it was important because the species was named from up there back in the 1980s.
Right.
And then you go
he's swimming
collect
away.
something that you think is the same thing, but you're not absolutely sure. But getting it from near the type location was really important. And one of the outcomes was that several people have been calling something from the Tonga area the same thing, but it turns out not to be.
He just swam onto the porch there.
Yeah, they wants to come with us, right?
Yeah.
Yep, they can be very good swimmers with their little feet down each segment are fairly large, so they can start using those and then an undulate movement.
He's being kind of cooperative. Volunteer.
What's really interesting about this work is sometimes, you know, a single specimen can be that you just think is, oh, it's in the debris of what you collected, and that's where really careful curation of samples, doing your best to figure out what they are, and then those samples being available for somebody else to, oh, what would be crucial is to understanding this from this place, but who's got the specimens and where do we find them? So if they go somewhere where you can search online to discover that Scripps or the National Museum of Paris or the Harvard Natural History Museum, guess what? Their online collection says we have something that
Did you just
has this
suction it?
name or is from
Nice
this
job.
site. Maybe it's got another name, but we can request that specimen to work on.
So, do you have the jar camera? Oh, you do.
Yeah, it's here. I'll give it a minute and.
And that was nice suctioning.
Yeah.
On the fly. He was being a little elusive.
Yes.
I think it's in the jar. I don't.
Yep, there it is.
Oh, nice.
Oh, yeah, okay,
excellent.
And then rotate that baby.
Yeah, rotate that baby.
Oh, thank you so much. Jar number.
Oops, I'm going to go the other
Jar
way
number
to the
3?
next jar.
Should
Yeah,
be
that
jar
was jar
number
3.
Yep, there he is. Okay.
That's got
oh no,
shrimp in
that's
it.
a shrimp
That's got
jar.
That was probably a shrimp that was in the hose.
Oh, okay.
Yeah.
Came in
Okay,
with the,
but there's the polynoid. Yeah.
Yeah. So we're
okay. Extra
I'm going to leave
shrimp.
it on jar 4.
Okay.
Nice.
Thank you. Thank
well
you
done.
so much.
We know there'll be scientists ashore that'll be excited about bringing home some specimens like that. And I also want to give a shout out to, on our live feed, joining us from Saipan, we have the Center for Living Independence group at the Mariana Trench Marine National Monument. So we're so grateful for the community joining us here and rooting on Nautilus and all of you here on deck. And, you know, there are scientists and there are elders and there are people from the community in the region of Marianas excited about this work and so curious to learn more. So thanks for following along, and shout out to Ranger Jihan at the Visitor Center in Saipan.
Yeah, that's great.
Yep.
Sean's in the room with us. Say hi, Sean.
High back meltdown. So you crashed your computer. Neither.
Yeah, half a day, everybody. Back home in Saipan. Doing my best to, you know, work with these awesome scientists and getting some samples so that we could share back home for our visitors. So, yeah, I'm excited about providing something to look at. That's not a picture or video, which are still pretty good, but you know, having the live sample in full scale was pretty good. It's a good way to experience these organisms.
Yeah, we appreciate, you know, having Sean here, and Sean as part of the Mariana Trench Marine National Monument region that we're in right now, the Todo caldera, where we're diving at about 2,900 meters. This is a unique region as part of that monument in the Mariana Arc of Fire National Wildlife Refuge. and we're grateful to be able to explore in the waters of the Marianas, and so glad that you're joining us here as we get a little bit more of a view of those anemones and squat lobsters free diving.
Yep. Thanks. I feel like I'm finally getting the hang of it.
All right, Dan, so we're continuing upslope. Okay.
This is my friendly reminder that if you're moving your microphone around, do mute yourself.
Roger. Sorry.
It's like a friendly reminder to the whole van.
Brianna, we had another question about the samples and whether they are preserved on the ship and distributed to various institutions, or how research is sort of shared. Nautilus is, providing this for the public, and so all of this is public, both the live stream, and then you want to talk a little bit about the sampling and how that's shared?
Yes, so all the organisms are preserved in ethanol, and then they go to the MCZ, or sometimes there's a few scientists on board who have a particular research interest,
Okay,
so they
copy
might
that.
take them directly back to their institution on loan from the MCZ. The geological material, it goes to another institution, which I'm not exactly sure which one. Do you know,
It's
Jacob?
URI.
Oh, it's URI?
Yeah.
Okay. And so all the rocks and that go to that institution, and everything, once it gets shipped back, which is usually shipped in the best possible way to avoid any sort of damage to the materials, the curatorial staff and everyone in the museums kind of make sure it's up on the database as quickly as possible, so the public and scientists can look on the database and see what's available and what was collected.
Oh.
And then they could loan it to work on their own research.
Yeah, and Jacob, at the institution where the geologic samples are stored, is that a similar setup where researchers, if they're interested, can request them for loan?
Yeah, I believe so. The rock library we have is called MGSL. It's on our URI's Bay campus. And right now, we're kind of in the process of moving a bunch of those rocks, which really aren't supposed to be moved. So I'm not sure if they're probably open to the public, but very limited because of all the complications going on with MGSL right now. But
I'm gonna jump in there just for a second. I feel like we've been here before. We have. You want to keep going same direction?
My thought was go up to 2935 meters depth and then contour around to the northeast from there.
Okay, 2935 and then northeast.
Right. Try and stay around that depth and contour around the side of the ridge.
Right.
Because the vents they sampled were about at that depth in the previous cruises, so we're gonna see if we can find them by kind of going along that contour.
At that depth. All right.
Yeah.
Push in just a bit, Aaron.
It's having problems. Yeah.
We have our top navigator working on it, and probably a data engineer as well down in the data lab, if you're referring to HiPAC.
Yeah, Ed, could I get, since the HiPAC is not functioning, could I get Dan's nav screen
Give
over
me
here?
a second.
That's Aaron up there.
Oh, I'm sorry.
Oh, you're good.
Yeah, we're just having a few HiPAC issues right now.
Yeah.
But
Do we know what kind
that's on
of
it.
fish that is?
Some kind of, they all look like rat tails to me.
Sadly, I'm not very good on fish, and we see it around a lot.
There's Dr. Drazen when you need it.
Yep, exactly. So yeah, I mean, it does look like a mackerel, but
Okay,
Yep.
you can go white, thanks. I harassed the fish enough.
All right. Who needed things changed? Was that you?
I did have a question on how the chimneys are preserved.
Good for another 20, I think, Anna.
They're just dried, but pretty sure. It also depends, like, I know the chimneys that we just got are like probably sulfides, so they're a bit more robust than maybe something that's anhydrate that can dissolve pretty easily, but not easily, but can dissolve.
yeah, you have to be a little careful. The guys that I see sampling them don't even want them washed because then the fresh water starts to alter the mineralogy. So they just air dry them on the deck, usually stumbling over them after a while.
Hey Dan, I was kind of monkeying around with the camera trying to get that fish on the screen. If I press the center button on the stream deck, would that reset it?
No.
Oh, okay.
Okay, what can we do here?
Yeah, it's fine for the moment. You were monkeying with the HD camera?
Yeah. Trying to get that fish on the screen earlier.
Yeah,
Fly the
it's
ROV,
bacterial.
not the camera.
Yeah.
Fair enough.
So that fuzz that we see on top of those rocks is bacterial. It's some sort of, it seems like it's all over.
Yeah, and it is a food source for these little crabs,
So,
which are more eclectic in their food requirements. They can eat any kind of organic matter, or something dies, they'll be scavenging it.
They don't seem to be too worried about competition with each other. They're pretty happy sharing this space.
We have seen one site on this ridge where they're literally piled on top of each other. You could see where space competition probably did come into play, and a few of them were missing one claw, so a little
is Bill still in here?
bun fight going on.
So northwest is, I don't see anything on my sonar. It's almost like it's downhill. So
okay, we might.
It's going to be hard to get to
right.
2935 going that way.
Okay, well let's turn northeast then.
Yeah, uphill's northeast.
Yeah, thanks.
Which we've been in that area before, but
yeah.
Yeah. We can kind of skirt to the north of where we've been and make uphill. So northeast maybe?
Yes.
Okay.
Bill, can you see the nav-G screen?
Yes, thank you.
Okay.
Good.
Am I pulling you? Yeah, I'm pulling you.
Yeah.
Yeah, you can come down a little bit.
Looks like there's a little bit of matte on these rocks, huh? They have sort of a little white
Yes.
slide back
fuzz to them.
around. So I've pulled you 180, and it should come back around. But when it does that, sometimes it tries to go the other way, and then it
Yeah,
can
and it
never
may
get
be
there.
wafting fluids rather than something coming through the
Right,
rubble.
right.
Can bring your head around to the northeast. If you want to, Anna, I didn't catch if you were moving that way or not. We're good. Yep.
20 meters to the northeast?
Sure.
Yep.
Yeah, I can kind of hot dog it out, and I kind of know we've been here before, so it's somewhat familiar neighborhood. Ah, that's what's happening. I get fooled by that all the time. So I'm actually right here right now, and what the clue was is Atlantis looking that way. So if you want, you can do a DVL reset. What's that? Yeah, I won't have that on unless you rarely have it on. But that green radio button right there is the one you're looking for. Thank you. The other clue
Here's
is the
another.
smear of blue dots
Sorry.
is the USBL, and it gets offset from the yellow, which is the DVL. And you think you're here, but you're not here, but it's, you can work it out by, I was like, why are you looking that way? You should be looking at me. So you can bring your head a little more to the north. There. I'm assuming they've pushed the dive out.
Yes.
Sweet.
At least to 1600 now.
Awesome.
Recovery.
Yeah, for those just joining us, we are on our first expedition of the 2025 season, and this is our first ROV dive. We are in the Toto caldera, and we are in the back arc of the Mariana region where we are exploring.
No.
Oh, I'm sorry. I am talking
the
too
back
fast.
arc.
We're about
We're
5 miles
we
south.
consider this an arc volcano, yeah.
Thank
So
you.
this is where the two almost meet. They're, further north they're way far apart. In the south they all come together. But Toto we consider as part of the arc, partly because it's this sulfur-dominated system, which we don't see in the back arc. So, yeah, we're near the back arc, but we're in the arc right where they come together in the south. Sorry to interrupt.
No, I appreciate it, Bill. That's very helpful. That's I need clarification too. So,
set your head for 045 for a minute for me.
Bill, do you want to give a little overview of the dive so far? We are
bring your head to 045.
Sure. Yeah, we're in the bottom of this caldera of this arc volcano. From this, we're looking for high temperature venting from this hydrothermal vent site that was discovered by Japanese scientists in the year 2000 and then visited again in 2003. The area hasn't been visited since then, so we're trying to return to those vent sites and do some sampling. We've been having trouble finding the the high temperature vents.
Trying to get a look at
Earlier in the dive, we
the
did
depth he
find
wants
a
here.
place that was venting clear shimmering water at a temperature of 102 degrees Celsius. So we were hoping to find, the Japanese cruises found some vents up to 170 degrees.
Let's
And
try
that was kind of a
030.
white smoker
015 somewhere in
fluid,
there.
so we were trying to find that.
We'll kind of go to the north here.
We think we're in the right area because there's all this white cloudy water that's at times makes visibility very difficult. So it's kind of a little bit of a catch-22 where the closer you get to those cloudy vents, the less you can see. So we've kind of been doing a search pattern, and now we're gonna come up to the depth where those vents were seen and then traverse along that depth on the side of this ridge and see if we can encounter them that way. You know, the positions of those vents from the previous cruise, there might be a little bit of a navigation offset from their positions to ours, so we haven't been able to find them that way. So sometimes finding vents is harder than other times. This is a little bit of a needle in a haystack situation for us. But along the way, we're also collecting some representative samples of some of the hydrothermal vent communities, the animals that live at these hydrothermal vents. And we'd like to be able to get some samples of the high temperature vent fluid if we can, too. That's one of our goals for this dive.
One of the ways that we have found works best, especially when you're working in a changing environment like volcanoes, is to carry markers with you. These have been invaluable at Northwest Rhoda, which is erupting and everything's changing, and then you can go back and be sure you're in the same place because there's a physical marker there, or the marker is completely gone because the mountainside is gone. The Japanese did leave 4 markers at this location. I've been hoping we would encounter them,
Yeah.
but again, that was 20 years ago, so it's possible whatever material was used has decayed. And I have to say, I forgot about putting a marker, getting a marker on the dive so that we could have left one, so that, because there is a cruise coming back here in December. Dan, I assume that you guys might have markers that we could use some other time?
Yeah.
Anyway, we could probably rig something up. Yeah.
We have, yeah, we have bucket lids.
Yeah, that's something we could think about.
We have floaty bits.
In case.
We also have, I haven't tried yet, we have some 3D printed material that is supposed to float.
Ooh.
How long it will float.
Oh, okay.
I've been meaning to bust some out and try some fancy markers.
Oh, well, wait till you see Bill's fancy markers at Northwest Rhoda.
If they're still there.
Yeah.
I guess a bucket lid's the old standby, is it? They float until they get
moved
overgrown.
on from that, yes. Syntactic foam.
Yeah. I don't know what's in our toy box. We purged quite a bit of we collect a lot of it during ONC, and then we purge some, and then there should be something in there that floats.
It was really easy to get spoiled having a nearby home port for a long time.
Yeah. Good for another 20.
Another 20 at 030?
Sure.
Okay.
I like that target there ahead of Atalanta. We could probably keep it moving, Anna, for the next 20, 40, 60 meters, maybe.
Okay, so we're at our target depth. So now we can kind of contour around to the northeast-ish.
Yep.
I guess we're already going that direction, more or less.
We are. I see there's a little hint of something here. Sorry, here, which is 60 meters away. No, we've 10, 20. Yeah. I don't know if we've been there or not. Oh my God, that thing is slow. Yeah, we've been ish there before. Nope, I'm stretched out. You got 15 under you, so you're good. You could come up 3 meters if you wanted to. Keep it out of the teens, that'd be safe. Yeah. That way we'll get there a little bit faster. I'm gonna turn off those downlights and strafe the gauges again while we're waiting. Yeah. All good still? Have you written those in the log lately? Yeah. Let's do it in the electronic log, Mackenzie. Or you can write, you can jot them there and then transfer them.
Looks like we have a kind of a calmer basalt that maybe broke off.
Yeah, it's a long piece.
That would fit in the bio box nicely.
Yeah.
While we're waiting on Atalanta.
You won't be light anymore.
Yeah, we're really late.
Do you want to pick up rock while we're here?
Sure.
Okay.
I think that'd be good.
Maybe not that giant one.
Oh, come on.
Yeah, well.
It's not that big. It just looks big.
We all have dreams, but.
It's only, it's like less than a foot long.
It's like the wiener dog with the giant stick.
Here, let's try to go for. Oh, well.
Oh my God.
I'm waiting for you to
Yeah.
point out a rock.
It's your chance to telestrate.
Maybe like right behind it. I was thinking kind of like that one.
Roger.
Let's see.
You want to turn the downlights back on for us?
It's nice looking.
Oh, that was a great guess.
Oh yeah. Perfect. Let me focus on that real quick. got some great images on the still cam.
Roger.
I think we're good.
So we've got some rocks in both boxes, right?
Open
Yeah.
the starboard box for me.
All the rocks are in the starboard box.
Yeah.
Okay.
The
So I guess
chimneys and the forward.
some of the what was in the scoops went in the forward. I
Oh,
guess,
yeah, that
but
was.
those weren't really just rock samples.
You
So,
want
yeah.
to switch the cameras too for me? So I got one in the front, one in the back. Go for one in the middle. Yeah, happy with that. That box. Well, that one's in between. Okay, you can close it up. That rock is bigger than I first imagined. Heavy too.
So for my geologist friends in the control deck, what is the process for samples that are on the ship? What are you hoping to do while we're out here, and then what's the plan once they're back home?
I don't know about what Bill will say, but I was kind of hoping that we could break out if the rock saw is on board, break that out and cut some of them open and see how they look on the inside, and cut them into thinner slices and kind of split them up into different institutions that may want them.
5, Mackenzie. So let's make it 10.
Bridge now. 30 meters at 030, please. Thank you.
The rock saw was on board just a couple weeks ago, so it's still good.
Bill, we have the high pack screen back if you need. Yeah, with the contours.
Okay. Guess I'll stick with the.
And let's change
the
our
pilot screen for now.
Change our course over ground to 060.
060.
Yes, please.
Okay.
Trying to stay at that 2935.
Okay.
Thank you, Dan. Not many, well, I guess there's shrimp, huh? I was just going to say not many squat lobsters, but that seems like we're on the periphery, not too close to any venting, but there's still a few everywhere.
But we've, like I'm pointing in a direction where we've extensively been.
Can we get an Atalanta light on, or at least turn it towards the light?
The barnacles on the rock.
Any light?
They're stocked barnacles, these.
Yeah. Yeah, not sure what happened there. Did you turn those off, or did they go off? Strange. I don't know which ones you had on.
I think we had them off for a while, and we had Herc in sight. So we saw it off in the distance, so no big deal.
Right.
But then once we either turned or you puppied off somewhere, Dan, then we just had a black screen.
So. I'm gonna try coming to the north here when I got time. See if it dropped down the other side. Because we've been there.
More of those barnacles, huh?
Yep.
More squat lobsters.
All good to take off here. It must be the pressure wave from Herc that gets them going.
Yeah, I can feel and hear me coming. Especially when we're hot dogging it.
Mm-hmm.
I was trying to see if I could drop down on the other side of the feature here, but seem to be coming up. There's smoke. Been there before. Seems to be the direction of the smoke. So 40 meters is that kind of feature. So 10, 20, 30. Yeah, we've been on that one before.
Kind of where that contour bends up.
Yeah, I think so. Yeah, I think we've been there. Let's see which way the current pushes Herc here for a minute, because I keep seeing smoke, but when I fly through, I can't work out which way it's coming from.
You know, it almost looks like there's some juvenile tube worms, you know, small tube worms that have been settled and beginning to grow up here.
Oh, yeah?
There's quite a diversity of animals. Like there's a large, I think, in the foreground, I think I'm seeing
What's that?
a different limpet, Bathyacmea probably, but don't know. And the barnacles are here. These are stalked barnacles.
Where do you think you see the tube worms?
Well, all these little things sticking up, the small ones. They're very short.
Like right in the middle where the lasers are? Or are those
of course,
barnacles?
I'm looking at a different
yeah, use
camera.
the telestrator.
So I've gone dead stick there for quite a while, and we're
this,
hardly
this,
hardly moving.
this.
Oh, it doesn't show up
So
here.
they're just little,
Yeah,
so
that's all right. I saw
no,
it.
okay.
Do you want to zoom in?
Are we waiting on the ship?
Yes and no. I kept seeing the smoke there, but I can't work out which way it's coming from, so I'm just letting her kind of drift in the current and
Could,
what little there is of it. So it's not like a super direction, but maybe
I don't know what camera this is.
kind of.
It's the main HD camera.
Yeah, that one is the still camera you're looking at.
Okay.
Yeah.
All right. I'm just wondering if there could zoom in on something that you don't need here.
Yeah, go ahead, Ed.
Yep, coming.
Full zoom there.
Beans.
Seems like we're drifting to the west very, very slowly.
Oh yeah, so they're sort of isolated, huh?
Which, the tube worms?
Yeah.
Yeah. Yep. But they're quite small.
Mm-hmm.
And I'm not seeing any red plumes on them.
No.
Oh. But these stalkers are the barnacles. You can see the stalk on the left side.
Yeah.
And they can move around on the stalk. They can swivel around and move into position.
But the bottom of the stalk is attached, right?
Yes, it is. So they can't move off that.
Uh-huh.
I was wondering if we could see them feeding, but because they have cirri that come out just like the ones you see in tide pools.
Yeah.
But I'm not seeing them extended. Many vent barnacles have bacteria on their gills, so they often just extend, sorry, their feeding appendages, they're called cirri. They're actually like tiny little shrimp on their backs with their legs sticking out, and that's what the barnacles feed with, is actually the legs.
Huh.
And so that's what you're seeing in tide pools, is the little legs coming out. And vent guys often have symbiotic bacteria, and they just hang the cirri out, but I'm intrigued that I'm not seeing them feeding at all. I'm not seeing them extending. But they would be alive, because they're this one has a juvenile, the guy attached to the stalk.
Right.
You notice those quite furry on the squat lobster here.
So the juvenile there would detach in order to...
Nope, they can't.
Okay.
Once they attach, they're stuck.
Okay, Ed, you can go wide. I'm going to have to turn and catch up with the Atalanta here.
Okay.
A fun fact about barnacles is that they're separate sexes, males, females, and they fertilize internally. So they have to touch.
Wow.
So just think about that as a stretch, you know, for these guys. So they do have to move on the stalks, but they can't walk. So to attach to the stalk of another barnacle, at least you're closer, right?
Mm-hmm. Getting a bit cloudy in this direction.
You're going to have to come up a bit there.
Actually, they may not be separate sexes, come to think of it. They may have adapted to being monoecious, two sexes in one.
There was a question too in the chat about the advantage of being on a stalk as a barnacle. Does it allow them to reach higher?
That's exactly it, yeah.
And so that water would be more disrupted closer to the rocks. Sorry, away from the disrupted water of the rocks.
For a second.
Yeah, I think it also, in this case, has to do with a lineage of barnacles that can cope with the vent conditions. We do find barnacles attached to directly to the rocks. Another group of barnacles further up on the arc. We'll see them at Northwest Rhoda. And they start out as juveniles with a very short stalk, and as the juvenile grows, it gradually comes down and attaches to the rock.
Weird.
It's so they're kind of intermediate in terms of the evolutionary lineage.
Okay, you can come up a
The
bit more.
old form is to have a stalk. The new boys on the block in terms of evolution are the kinds of barnacles we see in tide pools all around, where they're attached directly to the rocks. And it means you're not getting blasted by waves. It's a more solid attachment.
So is it fair to assume that these barnacles, this species, is pretty old?
The lineage is old.
Sure.
That doesn't mean that the particular species has a long evolutionary history. It just means the divergence. So that trait has been kept in that lineage, whereas this separate lineage has gone on and modified more and more and gone into intertidal habitats.
Sean, do you see barnacles in the CNMI in shallow water tide pool areas?
Definitely see them on the buoys that wash up from them in the open ocean. You know,
20
back
meters
to the shrimp.
east,
I was thinking about that answer I gave earlier. You know, we do have like shallow water shrimp, burrowing shrimp, cryptic shrimp. You know, there's some that clean other organisms. They're cleaner shrimp, so they do exist. We do also have those brackish waters, ones that folks, you know, sometimes catch and consume. But I do see barnacles on washed up buoys that wash back onto shore. They're not as common as you would find them in other areas, but yeah, they do wash up sometimes.
Lots of tube worms here. And it was pretty cloudy there for a bit.
Yeah, it feels like it's off to the east, maybe down the hill more.
Yeah.
We're too high here, according to your...
Yeah. Do you want to go back to that cloudy area and then go down slope a little bit, or?
Yeah, we're moving east now to drop down the hill.
Okay.
Sound like the right direction.
Sounds good. Yeah.
Not seeing any pink here.
This one's probably alive, but these are not.
Oh yeah, that.
Maybe in this hole here. Zoom in there real quick for us. There. Or Ed. Ed's over there.
Somebody's over here.
I don't know if those guys are healthy or not.
The tube worms, I'm not seeing many. I did see a couple with the red plume. Sometimes they just fool you, and they're pulled back in the tube, and so you just can't see through the tube.
I haven't seen any of their little red fans at all.
But no, I'm not seeing it.
Maybe that hole
I'm a bit surprised because it does look like
Gone all the way in.
it's like there's fluid coming out here.
Yeah.
See, I don't know whether this is somebody inside with a little bit.
Is that like? Is that part of a shrimp? Or?
Yeah, that's a shrimp.
Okay.
Yeah. Yeah.
Oh.
Okay, Ed, go wide. See if I can.
Yeah, they look kind of decayed around the tops, right? As if it's
Yeah.
I'm gonna
not nice and fresh.
Push past the toys and the housing.
keep sliding to the east here. It seemed like that was, because the current was pushing me to the west, so what very little hint of it there was.
We're certainly in the tube worm neighborhood here.
Yeah,
Yeah, they.
shallow, too shallow.
Oh, whoa. What are we at? 292.
Getting more sulfur on the bottom. Visibility's going down.
I can keep sliding to the east, and it.
Still might be a bit shallow, huh?
Yeah, we'll slide to the east here. It looks like it drops off that
Okay,
way.
good.
Let's do 40, mid-large, yeah. Get out here where we haven't been. It looks like it drops off to my right. Is that correct? We'll get down to Bill's 29. We'll try and drop down 10 meters. Maybe they'll follow the contour down here at the deeper target depth. It was smoky that way. You're going to be dangerously low there, Mackenzie. So we'll put Atalanta over here in the deep water, and then I'll try and come down, and then we can chase the contour line one way or the other. You're going to have to come up a little there. If we take a good heave there, you'd touch the seabed or breathe on it.
Kind of the edge of the sulfur area right there.
Yeah.
So we're looking northeast,
Yeah,
turning back around.
I'm going to turn back around.
Facing east now, southeast, and we're facing south. Yeah, so that was kind of the north edge of it, looks like.
Yeah. We've been in here a bit. I can't come down the hill anymore until we get Atalanta off to the east.
Well, we can just traverse around. I think, you know, when the visibility is poor, it's probably possible to go right by things that aren't very far away without seeing them. So
wandering around in a dark parking lot looking for something with a pen light.
Yeah, right. Still looking for our keys. We dropped them, dropped them here somewhere.
Yeah. We were down to single digits there on Atalanta, which is definitely breaking the rules.
In the delta?
No, altitude.
Oh.
I totally get how lat long between two different institutions or vehicles would not align. But boy, you sure would think that depth would. It seems like we've been following contour lines.
Okay, I'll drop down a bit, Mackenzie. I'm starting to get some altitude under you. but we'll keep it in double digits, eh? I'm gonna turn back around and look at the hill here. You'll have to come, you can come down just 4 or 5 meters. It's quite a good drop off here, right? Because I'm 20 meters below you, but I'm only 10 meters away. Come right down to 10 meters altitude.
We're getting close to that, the end of that 40 meters if you want to extend it.
Yeah, I think we should wait for Atalanta to make the move.
Okay.
If we move again, then it'll swing too far away.
Yep.
Then we'll be too deep.
Yeah, I think that's right.
Yeah, patience part of the game here.
It's a bit of a cliff right there, huh?
Yeah, frustrating that Atalanta's on the wrong side of it. Look down with your camera, Mackenzie. Yeah, look straight down. We're gonna break the rules here a little more. Sometimes you gotta
clusters of shrimp there on the sulfur.
You gotta double tap it.
Oh, just
sorry, yeah, I'm gonna come down a bit.
Yeah, right there.
Yeah, if you look down, if you can get that thing to look down a little more, we're gonna come down where you see seabed with it. So I'm at the magic depth here.
Okay, back at the magic depth.
And
do you want to?
Oh, I'll try and move around a little bit here, Bill.
Yeah.
I gotta get Atalanta on the east side of me.
Okay. Just
he's 7 meters off the seabed.
Do what you can. Wandering around at this depth.
Yeah. Looks more promising down here.
This looks like kind of rocks cemented in a sulfur cement.
And come down a couple more meters.
Lots of little shrimp everywhere on the sulfur. Some of those fissures.
Yep, we're getting close. We should be able to keep coming down as you slide off to the east there. I'm gonna.
Oh, are they? Can we go back and have a look at those tube worms?
Sure.
To the left?
Can hold what you got there for a minute. These guys here?
Yeah.
Yep.
Push in just a bit there. A little more if you want. I think I see a fan.
Yeah, they are alive. They've pulled in since you can just see the tips coming out. And actually that would be a nice sample because it also has a variety of snails on them.
On the tubes,
yeah.
Sounds good.
So we could get, a little scale worm there.
Right, that little red
these
guy.
Yeah, these look like provanid snails. A couple of limpets.
Yeah. I'm going to float up here, and I'm going to stuff a few of those right into the digital still camera.
Oh yeah, you can see the red plumes there.
We'll get some close video and should be able to get some
Look
pictures.
at the regrowth from here to here. It looks like something in the flow has almost reinvigorated growth. So when they get really more transparent, they're growing faster.
Oh.
Someone taking pictures back there?
Yeah, if we can get a still cam shot if you guys wouldn't mind, like scooching up a bit.
Yeah, I'm working. You'll have to hit the focus there and then take some shots.
Dan, when we're all done with sampling and whatever else we're doing here, I'd like to drop down and try and peek under this.
Can't quite hear you, Ed. You're real quiet.
All right, got a couple.
I can
got a couple good ones. So.
Roger. Roger. I'm just kind of waiting for Atlanta to stop moving, and then we'll park it. Hopefully you got a couple good pictures
so these tube worms usually come out a little further from, so they can expose their gills, which are the little red tips. Which camera there? People are, no. Which camera? is broadcast. So it is that one, yeah. Okay,
Yeah,
so
that
yeah,
means
you can
this.
see the red tips and the gills, just as I was describing before for the clams. The same thing happens here. The gills are taking up the hydrogen sulfide, the oxygen, but in this, and the and the carbon dioxide, it's been a long night, carbon dioxide, oxygen, and hydrogen sulfide. And then those compounds travel in the blood, but the blood again is ensuring, the blood hemoglobin is ensuring that there's no reaction between the sulfide and the oxygen until they can deliver it to the trophosome. Now the trophosome is what takes up the majority of the tube inside. It's a special organ that is evolved in the cyboglidum polychaetes, that chemosynthetic systems. The trophosome houses all of the microbes. And then the dissolved gases are delivered to the trophosome through a highly intricate vascularized blood vessels system. Bacteria take it up, do the same conversion to get energy, and then creating food products in the form of glucose, which then leak out of the cells to the adult worm. So the more dissolved gases that the worm can deliver to the bacteria, the more food it gets back. And then they grow faster.
You can see drop down a couple
But
meters.
you can see these worms have all grown over the little fissure edge here because the fluid's coming up from below.
Right. So they'll just hang over, and then when they're ready, they'll pop out.
Yeah.
Their gills will extend, and they'll bring in all that nutrients for their... And so the red that we see on the inside of the tube, that's the length that they'll extend out?
Actually, this species only has a very short plume. It doesn't grow really fast. It's not kind of like the iconic Riftia, the giant chew worm at Galapagos with the really, really big plumes. This one is adapted to living in very low flows. You wouldn't find Riftia living in flows like this. Instead, these worms can extract much lower concentrations and don't have such a large body mass they have to maintain.
You want to switch cameras?
And how about those snails that are taking up homes on the outside of the tube?
And they're grazing. Yeah.
Picking up anything that the tube worm misses.
And there is, well, the tube worm is only taking up the gas, the three compounds, the gases. The grazers are grazing bacteria. The tube worms slough off organic materials. Sometimes that settles, so the snails will take what they can in terms of organics that they can pick up.
What was the species name for this tube worm?
This is Lamellabrachia juni.
Juni.
J-U-N-I. It's named after a Japanese scientist.
Okay.
Let's move.
Were these discovered or described in 2003 during those?
No, it was already known from a site near Japan.
Yes, please. Yeah, I see. Sonar. Okay, and you can try and tighten up again there. You have to open that box all the way. Are you guys ready to grab a handful of these?
Yes, sir.
Here we go. Come wide just a bit.
So, Rena, are you just seeing snails or snails and limpets? Or?
Snails and limpets, and I did see a couple of scale worms.
Yeah, I see one in the lower left there. Red guy.
Let me come out to the arm.
What's that?
Yep.
Let me zoom out so I see
So you notice that
Yeah.
right down at the bottom left, there was a little red
Oh, there you are.
scale worm curled around the top of the tube. These scale worms are predators,
Oh,
and they
yeah.
have a jaw that shoots out. You remember from Axial?
Yeah, yeah.
Yeah. And they lurk at the top of the tube, so when the tube expands, they creep slowly up to the edge and then suddenly shoot out this set of jaws that can come out.
It's a little bit like Alien.
Yeah. Come out almost half the length of the little scale worm.
Yeah.
And they got 4 jaws on the end, and they take a bite out of the...
It is like Alien.
You guys, you happy to grab there, Brina?
Yeah,
Or
let's see,
more
is that...
back in the root?
Actually, that should pull out all right.
Yeah. Zoom in just a bit there for us, Ed. You reckon?
Yeah,
Okay.
give it a try.
I'm drifting a little bit there.
Yeah, the roots are pulling out. That is excellent.
Okay.
Now see how stiff they are. And I have to tell you, right there is a lot of time in the wet lab, because we have to get them out of the tubes.
You happy to put those in the box, are you?
Yes,
That's a
I
yes.
think that's a yes.
Yes. Yep, perfect.
And
I
is
hope
that
they
enough?
sink.
Yep. And
You sure?
I think it's fine for the
Okay.
people that want it.
So we're going to be pulling out the worm from the tube it's in,
Well,
in the wet lab.
we aren't pulling it out. It breaks,
Okay,
so we have to
close
cut the
box
tube
a little
open.
for me.
And that's going to be difficult because
And-
it's so...
And sometimes you
going.
can blow them out from the other end.
Oh.
If you've got a good set of lungs.
I don't want
Anybody
Mike
in the
and Ed.
lounge that's feeling
Turn that light on.
particularly strong in the lungs today? Got something for you.
I got it.
Oh, man.
Okay. Hold that. I'm going to let go. Okay.
There are
close.
not a lot of specimens of this species around in the world, so it's nice to have them available.
And you were happy with just the one grab?
Well, what, you've got about 15, 16 in that handful?
It looked about that many, yeah.
Yeah, I was not counting. But I've slid off here. And yeah, sure. Not sure what the delta is there. No, you're good. Let's we'll get sorted out here. So you have to turn. I don't know, maybe clockwise. You can turn back around and look at Herc. And I should be able to turn around. The other choice, which way you turn. Yep, keep clicking.
Look how they're all draping downhill.
Yeah.
We had a question in the chat about the buoyancy of the tube worms. Are they neutral, positive, or negatively buoyant?
Probably slightly negative.
You can, yeah,
And does that have to do with their orientation here on the...
No, they're stiff enough they can grow. They know what direction they want to grow in. if you'll see them growing up, if there happened to be something up here, they would have gone the other way.
Okay, we're still at the magic depth, and all of our ducks are somewhat in a row here.
Okay. Yeah, let's explore the magic depth.
All right.
Which, one way or the other. Do it one way and come back and do it the other? Or?
Sure, yeah, I can move around a bit here before we move the boat and make a decision.
Yeah.
Or vice versa.
It's like an overhang, huh? Is there a kind of a fissure below us?
Herc really wants to fly under there and find out.
There was a bit of a...
Bill, you want to talk about how a fissure forms for those of us that are geologically challenged?
It's just a crack. Yeah, you know, I'm struggling myself, just kind of visualizing.
Bring your head
this.
to the right a little for me, Annabelle, and
we're on a
come down
pretty
a
steep
meters.
slope, but there's places where maybe parts of the ground have slid further down slope or something like that, or maybe this whole ridge was kind of pushed up, and so there's cracks where that was kind of buckling or doming upward. Yeah, pretty, I mean, it seems like we follow the slope along for a ways, and then suddenly there's a big cliff, and that seems to be where, you know, it's spread a little bit, so a crack has opened up, and the sides have moved apart a little bit, so.
What's the chances of it caving in and burying me?
Low.
Low, right.
Yeah.
We're definitely under it right now.
Yeah,
Can't quite see it at
I wouldn't go ramming anything, but.
No.
Is there any connection between the hydrothermal vents, the location, and these ridges?
Yeah, I mean, it seems to be the vents seem to be along this ridge and near this fissure, so I think that's part of how the hot fluids are getting to the surface.
Check out Herc sonar.
That's kind of a general concept that don't know exactly how that works, but yeah, I think there's
barnacles.
Yeah, lots of barnacles on that overhang that are reaching down. Just looking at those barnacles on the overhang.
It's miles away.
Yeah, so we just moved from tube worms to barnacles, and so the barnacles don't need so much fluid flow. That looks like a little sponge, doesn't it? Yeah. Up over the edge and microbial mats.
Yeah, obviously they like the edge of that fissure where there's
some stuff coming up.
Yeah. Water wafting by, and they can filter feed things going by. Yeah, they're really happy there.
They are. So we've dropped about 10 meters and gone, I don't know, 10, 15 to the north there. Following the feature.
Yeah, and are you kind of getting towards the end of your leash, or?
Yeah, maybe another 5 meters here, and we'll zip back the other way.
Yeah.
Oh.
It's amazing, huh?
That is impressive.
Yeah, there's so many of them.
I have to say it might be a nice idea to have a sample of that at some point.
Might as well do it now.
Right, let's see if I got enough leash to peek around the corner.
Do you want to try to get a rock
that
has barnacles on it?
Yeah, like something like that. Yep. That way you're not breaking breaking them.
Right.
Yeah, I dusted it up there a bit.
Plus you get a bonus rock.
I was working pretty
Exactly.
hard to pulling my tether around. If I come back and put our 6 on Atalanta, I can pluck one of these here if you want.
Okay. So we're going for a rock with barnacles.
What's that?
Going for a rock with barnacles.
Sorry, say again, Mackenzie?
Oh, sorry.
Roger. Yeah, I think you're all right. Everything's static. We moved you back a little closer to the danger cliff there. Am I hitting?
Did you want us, did we get a squat lobster in one of the sections? And did you want?
I don't think we've gotten a squat lobster yet.
No.
We've only gotten shrimp and that one polychaete.
Do you want to try to section?
Ooh, that's got a parasitic isopod. Yeah, well, it would be a good idea again to confirm the species. I can only guess from looking at it, and I don't believe they were collected there. Not on the list.
I'm gonna have to slide back around to the south there. This is not working for me.
Yeah, there's plenty of barnacles, so we could go south.
Yeah. When I turn like that, fight Herc's tether.
Yeah. It's trying to wag you all the time.
Well, yeah, I used the wrong thrusters and thrust up against the cliff. I turn the disruptors on.
Well, we might have to look up for a loose. Where barnacles are and where loose rocks are, not necessarily overlapping here.
They were, historically, anything I've touched has been loose.
Don't pull the whole wall down.
I think that one will come loose right there.
Yeah.
I like that one.
Yeah. And then if we could suction a squat lobster or two at the same time, that would be okay.
Oh, yeah.
As soon as he touches, they're gone.
Oh yeah.
So far it's been squat lobsters many, Dan 0. There's a really funny video that's been shown over and over. Epic squat lobster fail. I thought it was being so sneaky. Outsmarted by a lobster.
I'm looking at these ones over here too. I don't know if the setup is as good there, but
yeah, that's fine. Well, I might have to move. You want to get a tight shot of the rock before we volunteer, Ed?
That'd be good.
So Rena, you would say these, that barnacle's got
look, see the cirri are extended on that one?
Yeah.
Sorry, I'm moving a bit there.
These barnacles are not hydrothermal species.
Yes, they are.
Oh, they are.
Yeah. Yeah, barnacles tend to live way out on the edges, kind of like at Rhoda where they're off there on the
periphery.
Yeah.
So how is it that they're hydrothermal dependent?
They are growing microbes on the cirri.
Oh, I see. Oh,
so
wow.
they just leave the cirri hanging out, and it grows the barnacles, and every now and then they lick them off, put them
Grows
out
the
again.
bacteria.
The bacteria,
Right.
Grows the bacteria.
Wow.
These ones are kind of, I suspect it takes a while, but I've seen some where when they extend the cirri, it's just like a giant fan with all this white stuff coming off it. These seem quite
Okay, push in
sparse. I mean, we were just looking at the cirri. Can we just take a look first before you do that? I'm sorry.
Yeah, let me move my weapon out of the way here.
Do you want to
just pan down? Yeah.
Something you wanted to see closer there?
Yeah, okay, see this barnacle right over to the left?
Yeah.
Oh, yeah.
And it just expanded its cirri. They're kind of thin, but
very thin.
unlike barnacles that you see in the tide pool that are always moving, and because they're trying to get stuff out of the water, he's just leaving it out there.
Yeah.
Right?
Go left. Oh, there's a couple of them.
Yeah, that's right.
Boy, they're so delicate.
Yeah, so, look at them all coming out. I mean, this is quite lovely.
Sorry. Herc just decided to find another parking place. Go ahead and zoom in there, Ed. Which one were you looking at?
All
those.
That one just retracted.
Yeah, everybody
Oh, and they're coming back out.
got a little nervous there.
That one on the right is back out. Yeah. A little more stuff in the water. Huh.
Yeah, sorry, Herc sighted on a new parking place and wrecked the viz.
Oh, yeah.
And I don't know how they feel about particulates, whether they're kind of pulling them in, and I'm...
Wow.
Yeah.
They're kind of curved on the end, huh?
Yeah, that's right.
And is it like a branching thing?
No, they've got tiny little bristles in between.
Yeah.
But they're just trying to capture. They're curling in. That's fantastic. Actually, I've never quite seen it like that.
Yeah, they curl so much. It's like a...
Can we take a high-def picture of these extensions?
Yep. I can try. Can't really see them. Oh, actually, would you guys be able to zoom with the still camera?
We're drifting further away.
Or is that?
Yeah, I think it's zoomed in now. Yeah, that's
right.
as far as it zoomed in.
Okay.
The porch is out right now, and
I see.
Yeah.
The porch view is not great.
I'll just take one just in case.
Dan, I'm having to continue.
Never know what you might be able to see.
Further away.
I can't hear you, Ed.
I think Brianna's got some good captures though, so I think we're good.
Yeah.
Yep. Roger. Yeah, Herc's doing what it's doing slowly. Okay, here we go. I'm gonna come off. Should come up just real nice and easy, a couple meters. We're just gonna we'll reposition here, and I'll go get in there again.
Okay.
Yeah, just as a reminder, that guy.
Got your eye on it.
Yeah.
Good.
So as it retracts and it brings the particulars back in, is it almost kind of like feeding into its mouth? I mean, how is the
Yeah,
process then?
there's if anybody online has ever done biology and had to dissect a crayfish or a lobster or eaten them, there's all these little mouth parts. And so this is like a little tiny shrimp or lobster lying on its back, the legs coming out, and when they come back in, they go past these mouth parts that scrape off the goodies and then come back out again. And the wonderful thing about arthropods is how they have diversified all those appendages to do different things. And so the mouth parts are, when I say mouth parts, they got mandibles, they've got maxillae, they've got maxillipeds, and pairs. Like there's 8 different pieces that.
Okay, Ed, good. Full zoom again.
Yeah, it's amazing.
Yeah.
I think Hercules could take a tip, try one of those arms to pull in some squat lobsters.
Copy
maybe.
it. No, not force stocking them. You can zoom in there again for us. You can zoom in, please. Okay. Hold that. Yeah.
Is that another one of those polychaetes?
Yeah.
I'm gonna pick up the rock, guys, and then.
Yep, that's great, man. Whoa.
Well done.
How well.
Nice snag.
Okay. Well, I think you'll know which
Tilt
one
up a
that
little.
was.
Yeah.
The one with the barnacles.
Mackenzie, tilt up on the HD. And, yeah, okay.
It's both. We're very efficient here.
What box do you want it in?
Starboard's fine.
Yeah, I think starboard.
Okay,
Yep.
you can switch
I've been
cameras and
making
open the
notes on
starboard
where
box.
they all are.
Hey, Data, what sample number is this?
12.
12, roger.
Go wide for me, Ed. Thank you.
Thank you. All right.
Here, let me show you my best side. Hold still, Herc.
Okay. I think one more angle of it maybe having the side that's kind of discolored. All right.
Are you good?
Yep.
All right, in the box.
Getting a lot of love on the chat from people watching from all over. We have a comment from Saipan, appreciate the work we do. Friends that have been on the live stream for many, many years and are just so glad to be back for our first ROV dive of the season. So shout out to the crew here and everybody and all the hard work.
Thank you.
Okay, that rock is second one back. 4 little rocks in a row. Where are you going, Herc? Herc, where are you going?
So, Rena, do we want to suction us, attempt to suction a squat lobster?
It would be great.
Okay.
We'll
We'll test
wondering.
Dan's skills in the squat lobster department.
Okay. Look, there's one right on the front of Herc.
It's a volunteer.
Take me home with you.
There's another one.
Let's see if I can show off here in the last few minutes before watch change.
Yeah, you have a chance to
redeem
rewrite
yourself.
A video highlight. Rewrite the narrative, Dan. You can do it.
It would be pure luck if I do. I'm milking it till watch change here, by the way.
Oh. This could be, not that one.
We gonna miss.
Didn't want that one anyway.
No.
Oh, I thought you were driving the just keeping the suction going as you drive the sub.
That's what he was doing, I think.
Totally.
Oh, he was. Okay.
Look at all these volunteers
we got
up here.
We got the polykeet that way.
We should have the next watch walking in the door any second. So
oh,
there
you got him. You got him.
Oh.
Yeah.
Oh, yeah.
Nice.
Okay.
All right. New narrative. Dan 1, squat lobster 0.
Actually, we're 1-to-1. That maybe makes up for my last
I'm just saying
fail.
2025.
And
we got a
rotate,
new scoreboard.
See which way we go.
Good.
Oops, wrong way. 4, so I'm going to rotate around to jar number 5.
Good.
And I'm going to retract my porch. Amazing that the majors are still tie wrapped in. There's a few bonus samples hanging out on the porch there. And I'm going to slide back to the south here and put it in the box for watch change. Mackenzie, you want to come up a little bit?
Data, was that sample number 13?
That sample, the squat lobster, was sample number, 13.
13, roger.
Okay, hold that for a second. I got back out from, we're underneath that overhang there, I think. And come up a bit more. Didn't hit. I don't know what you're talking about. Yeah, keep coming up. And can you guys, before you go, reset the DVL? Come up, yeah, keep coming up. That's something doesn't look right there. Look down and come up a little bit. Here, I'll come up. You look down. That should be back in the proverbial box.
For those following along, stick with us here as we undergo our shift change.
Front row signing off for watch change. Thank you, everyone.
And again, if you're just joining us, we are on our first ROV dive of the 2025 expedition. We are NA-171, and we are at the Toto Caldera, and as part of the Mariana Trench Marine National Monument. And we are excited to be exploring some hydrothermal vents and collecting some samples and doing some awesome science. So thanks for joining us, and we'll continue to be here as the shift change happens. Stick with us. Please do also share your questions with us on the live chat, and we'd love to get to know the crew here and share a little bit about the work we're doing here at UV Nautilus on NA-171 here in the Mariana in the Mariana Arc of Fire National Wildlife Refuge. Please share with us here.
Yes. Party line, party line.
Can hear you? Party.
It's a party on the party line.
How does
Party
my
line.
audio sound?
Science folks, you look like you're ready to introduce yourself. Should we do a little row of introductions? Kelsey, you want to start?
Nope, not on party line.
try one more time.
You could maybe check your aux volume for your mic. It is up. Try this little aux volume right here and turn it up and maybe see if that helps. Still doesn't.
Ooh.
Oh, you flip up on the mic mute? Up. No, it still says your thing is muted. Oh wait, are you doing...
Turn these lights off.
Oh no, you gotta do this one.
There we go. Party line test.
Nice.
Party line, party line.
Party
Yes,
line,
you are
party line, party
live.
line. Awesome. Hello, everybody. My name is Kelsey McClellan. Sorry for the technical difficulties. I am sitting in the data logger seat. Sierra, over to you.
Yeah. Hi guys, I'm Sierra Landreth. I'm a grad student at Florida State University. I am also one of the scientists today, and I largely study seamount ecology like deep sea coral and sponges.
And yeah, and I am Jeff Beeson from Oregon State University. I'm an assistant professor, and I'm in the watch lead seat. I'm a marine geologist and do geophysics as well. And I'm super excited to find some hot water to put in our fluid samplers.
Woo!
We're fingers crossed.
We've checked off a lot of objectives today, so I think we can definitely do that. I feel confident.
Get that fluid.
Front row, are you on party line, and would you like to introduce yourself and your role?
You want to go, Annabelle?
Yeah, I can go. Hello, everybody. My name is Annabelle Baker. I'm the Atalanta pilot for this watch. And I'm also a student at Oregon State University.
Michael Hannaford, the Herc pilot for the 12-4 watch.
Hi everyone, my name is Isabella Monaco. I am the ROV navigator from the 12-4 time slot.
Clarity, I'm up there. Nice.
So it still looks like we're seeing what they saw earlier, which is a bunch of non-living tube worms. I don't really see any color there, but all those things that kind of look hairy on the seafloor, I've heard some people say, those are tube worms, and they are found typically close to hydrothermal vents, or sulfur vents, or different kinds of vents.
All right, guys. So what was the handovers up there, front row, for what we're doing? We're looking around at this roughly water depth for venting sites, right?
Yes.
Let's see. How are we on a vessel move right now, or are we free?
We're free. So if there's any direction that we want to take this party, we can go there.
Bill had the navigation. Is HiPAC working again?
It is for the moment.
Okay, let me see if I can put HiPAC up here.
Right now we're kind of located around waypoint 8 from yesterday. I can zoom out to give a full perspective.
Well, let me see if I can get HiPAC to go on the KVM over here. There we go.
Just wanted to make sure.
No. Push down on SPL for talks.
There we go. Is it working now?
Yeah.
Yes.
Okay. So a little bit of technical difficulty. My name is Shannon Celine. I'm going to be your Science Communication Fellow today. We are just getting back on an ROV dive to the Toto Seamount, and we are in the Marianas Trench Marine National Monument. At this moment, we're looking for venting systems. Some of these areas have been mapped previously, and we are going to go and look for changes that have happened since 2004 when it was last explored.
Okay, I'm looking at the high pack, and so the way what they were doing on the last watch was targeting this depth rate, like this depth contour. So if we can just, let's move north around the same depth contour and look around for venting or, you know, large assemblages of tube worms and investigate them. We'll start north, and then we might move down contour a little bit, down slope, and then do to the south.
Okay.
Roger.
Thanks.
Yep. Yeah, you can put that in now.
Could you, Sierra, go back in C-Log to yesterday and give us search, if you can search, or look for where we had venting that we saw yesterday.
Oh, like look for the sites, okay.
Yeah, and if we could get those lat longs or where they were close to, we're going to put that in our back pocket if we need to just go directly someplace where we saw some more focused fluid flow.
Gotcha, okay, yeah. We can look for it. I think you gotta go to older events, maybe. We didn't look.
So front row, if you haven't introduced yourself, can you please introduce yourself?
We already did, I think, a little
I think
earlier,
I
but we
missed
could probably
that.
go again.
I missed that one, actually.
Multiple times.
My name is Marin. I'm the video engineer intern for this watch. I'm a student at Florida State College of Motion Picture Arts. And yeah, that's me.
My name is Isabella Monaco. I am an ROV navigator for the 12 to 4 slot, and I'm a contractor with Ocean Exploration Trust.
I hope it's working.
Michael Hannaford, Hercules pilot, and I'm also a contractor.
I'm looking around, sorry.
I guess it's my turn. Annabelle Baker, Atalanta pilot, also contractor, also student at Oregon State University.
Can we start with Kelsey on the back?
There you go.
Hello, my name is Kelsey McClellan, and I am a program coordinator for the Natural Resource Management program over at the Northern Marianas College. And I'm sitting in the data logger seat.
I'm Sierra Landreth. I'm also one of the scientists helping out today. I am a grad student at Florida State University, and I largely study deep sea seamounts, specifically coral and sponge communities that we see on Pacific seamounts.
Sorry, I did it. I got excited.
All right. Did you find? Okay, we'll use that in a second.
Okay.
Yeah, Jeff Beeson, assistant professor, Oregon State University, acting as the watch lead here from the 12 to 4 shift, marine geologist, and I said it earlier. I'm going to say it again. We got to find some hot water.
Hot water, okay.
Find some hot water.
Which means we need to find shrimp and everything that lives near the hot water.
Yes.
Yes.
They are our indicator. Where the shrimp cluster, that's a good place to go take a look.
So the vent is going to be next to Crustacean Nation.
So at 1840.
It will host the Crustacean Nation. Yeah.
So at 1842 yesterday we have Herc taking temperature probe of hydrothermal vent. Is that a good place to
Yes.
look?
Could
Let's go there.
you get the lat long?
Why I have the lat long.
Okay. Maybe what's the most efficient way to get this to our navigator? A piece of paper?
That works with me.
Okay.
Great. I love high tech.
Yeah.
Yeah.
We need some sticky notes to make some like paper airplanes.
I've got a sticky note if you need it.
Our carrier pigeon.
Whoa. Do you want to just
That's
chuck
the
me?
gauge check.
Yeah, there we go.
Thank you.
Perfect.
So for the future to do it, you go to
I don't know
review
about that.
cruises dives.
Oh.
Yeah. and
What
then
is
we
that
go
thing?
Into review. And then you go, I just typed in freeform.
Freeform it.
text,
Okay.
that's where I found it.
Awesome. Thank you.
Gonna come down a little bit here, give you some wiggle room.
Sure, yeah.
That kind of looks like a sponge almost, but
It's pretty big compared to the sponges we've seen.
yeah.
12, 7, 1, 2, 5,
Some
3,
kind
2,
of demo spongier. It's very encrusting.
Yes.
It looks like it's encrusting on the rock there.
What would cause encrusting on a rock?
So sponges have a lot of different kinds of morphology. This just happens to be one. So you'll see a lot of shallow water sponges too also tend to find rocks and just situate themselves on it as like an anchor, especially in areas with high currents and high wave action. So they can be encrusting, they can just be massive, they can be branching, they can be fan-shaped, they can be stalked sponges too.
What was our bearing?
So there's just a wide variety that they come in.
Okay. Roger. Isabella.
Yeah, we could add that point, put it as a waypoint or a marker on HiPAC and just see how far away we are from that.
And I'll
Sounds good. Give me 1 second.
Okay.
I'll also continue maybe looking up other venting, because if the ship is too far from that waypoint,
yeah.
do we want to navigate back? Okay, so I'll keep looking.
Yeah, I can find other
Might
spots
start
maybe.
coming up again once we do
Which
the move.
waypoint are we
Yep.
hypothetically headed
You're all
towards
right.
now?
Just hold that.
We're
just like you
we're
got it
kind
there.
of at
Hold that 20
waypoint.
right
We're next to waypoint 8, but the plan kind of started on last watch to take the water, the depth from the cruise report from the Japanese cruise report, and use that depth as a marker and just follow the depth contour. So we're going to go north along the contour, and then we're going to come down and go south. So we're near waypoint 7 and 8, but we're pivoting away from the waypoints because they haven't been working very well for us.
And right now we're at 2,936 meters.
Yep.
And our temperature is 1.8 degrees Celsius.
Yeah, cold.
Pretty chilly.
Here's another one.
It's the only thing colder than the control
Coming
van.
up.
It's actually not too bad this morning.
Feels okay.
No, it's not.
Feels okay right now. Yeah.
Knock on wood.
Then yesterday around 14:47 or 2:47 p.m., it looks like there is free text of shrimp and venting near shrimp. So I'll also pull that.
Okay.
Maybe.
Yeah,
Or
I
no?
think 2 o'clock would have been our shift.
Yeah, 2:45. This is Sierra Landreth.
Yeah.
That's you.
I remember us seeing some little openings with some fissures. I don't know if any of them looked as good as the one that you gave them already.
Yeah, that one should be priority.
But let's have that written down.
Okay. I'll put it second priority maybe.
Okay.
Yeah, if the first one maybe doesn't work out, we could always try the other ones.
So that first one's going to be the priority contour.
Yeah.
Cool. Sick. Venting near shrimp.
Gonna scoot up another
There's
snitch.
a lot of white rubble up there.
Yeah, I'll go check that out in a second here.
And this looks different even from what we were seeing on